So a hydrolase is using water. So with water, it catalyzes the cleavage of a bond. And an example of that, would be alcohol dehydrogenase phosphatase. Sorry. This is aspartate. Asp D. It's polar acidic side chain. Aliphatic. Acidic negatively charged. Asp. Glucophosphate, phosphoglucose isomerase. Joins two large biomolecules of the same, often at the same time. Cleavage of fatty acids. Hydrolysis of fats. kinase adds the phosphate group from ATP, but phosphorylase adds the phosphate group from inorganic phosphate. HPO4 minus. This is suicide inhibition is irreversible when the enzyme binds the substrate analog and forms an irreversible complex.
Irreversible inhibitors are molecules that bind onto active sites of enzymes and inhibit that enzyme's functionality, inhibit their activity. Now, the thing about these irreversible inhibitors is they can bind onto the active site either by covalent or non-covalent means, but once they bind onto that active site, they will not let go. And that means they bind very, very tightly, very, very strongly. And even if we remove access inhibitor from that mixture, that will not dissociate that inhibitor, that will not reform that active version of the enzyme. So all the different types of inhibitors, irreversible inhibitors that we have in nature and that we can synthesize in a lab can basically be categorized into three different groups, into three different categories. We have group-specific inhibitors, we have substrate analogs, also known as affinity labels, and we have suicide inhibitors, also known as mechanism-based inhibitors. Now, we can actually use these three different types of irreversible inhibitors to actually probe and study the residues found inside active sites of enzymes. And out of these three different types of groups, the most specific type of group is the suicide inhibitor, and the least specific type of group is the group-specific inhibitor. So let's begin by discussing the group-specific inhibitor. Well, the group-specific inhibitor is an example of an irreversible inhibitor that binds to and reacts with specific side chain groups of amino acids. For instance, on the board, we have two examples of group-specific inhibitors. We have idoacetamide that reacts with cysteine side chains, and we also have diisopropyl phosphofluoridate, or simply DIPF, that reacts with serine amino acids. For instance, if we take an enzyme that contains an active site, and inside that active site we have a catalytic amino acid, namely the cysteine, well then this idoacetamide will react with that catalytic amino acid, the cysteine, to form a covalent bond between this carbon here and this sulfur atom here. And because this is the sulfur atom, part of the side chain of the cysteine that is used to basically catalyze some specific type of reaction, because we form a covalent bond as shown in the following diagram, that essentially deactivates and inhibits the activity of that particular enzyme. And so as a result of this covalent modification, we essentially deactivate that active site of the enzyme. And notice in this process, we essentially kick off the iodide and we also break off that H atom. And so we form this molecule as shown here. The H has a positive charge, the iodide has a negative charge. Now, this is the other example, and in this particular case, we see that diisopropyl phosphofluoridate actually reacts with serine amino acids. For instance, we know that in the active site of acetylcholinesterase, we have these serine amino acids that catalyze the reactions, and so if we mix this active site, so an enzyme that contains an active site with a serine, then this diisopropyl phosphofluoridate will essentially form a covalent bond between this phosphor and this oxygen, and that will kick off the F, the fluoride, and also that H to form the following covalent modification. And so these are two examples of group-specific irreversible inhibitors. Now, let's move on to the second type of category known as substrate analogs, or more commonly, affinity labels. So the thing about these affinity labels is the structure of that particular irreversible inhibitor actually resembles the structure of the natural substrate that binds into the active site of that enzyme. So these irreversible inhibitors are molecules that resemble substrates, and this allows them to actually fit into the active site of that enzyme, and once inside, they essentially react in a covalent manner. They modify the residues found inside the active site in a covalent manner, and that inhibits that enzyme's activity. Now, what's one example of an affinity label? Well, when we'll discuss the process of glycolysis, we'll see that an intermediate molecule in glycolysis is known as dihydroxyacetone phosphate. And what happens in glycolysis is dihydroxyacetone phosphate is transformed into another isomer by an enzyme we call triose phosphate isomerase. Now, inside the active site of a triose phosphate isomerase, we have this glutamate molecule. And this glutamate molecule, the amino acid glutamate, essentially is responsible for this catalyzation process from transforming dihydroxyacetone phosphate into another isomer. Now, if we add bromoacetyl phosphate, this molecule, into the mixture, notice how this is almost exactly the same in structure as this original natural substrate. The only difference is, instead of this hydroxyl group, we have this bromide. And because of that, this will act as a substrate analog, an affinity label. And what happens is this carbon here reacts with this oxygen to form a covalent bond, a covalent modification. And that kicks off that bromide. In the process, because we formed the covalent modification, that essentially inactivates the active site of that enzyme, the triose phosphate isomerase. And now this enzyme cannot convert this molecule, the dihydroxyacetone phosphate, into its isomeric form. And we'll talk much more about that in our discussion on glycolysis. Now, let's take a look at the final category, suicide inhibitors. So these are the most specific type of irreversible inhibitors. And what that means is we can build and we can use these suicide inhibitors to basically bind to specific active sites of specific enzymes. Now, suicide inhibitors are also known as mechanism-based inhibitors. And that's because these suicide inhibitors, they can actually bind onto the active site of that enzyme and begin the normal catalyzation process as if this was a normal substrate molecule. However, down the line, somewhere down that pathway of that reaction, what will happen is that suicide inhibitor will produce a reactive intermediate that will modify the active site in some covalent way. And once that modification takes place, that inhibits that enzyme irreversibly. And so, two examples that we commonly use in medicine of suicide inhibitors is penicillin and aspirin. So remember, in our discussion on irreversible inhibitors, we said that penicillin is essentially an antibiotic, and this penicillin molecule binds into the active site of a specific type of enzyme used by bacterial cells to build bacterial cell walls. And this enzyme is known as transpeptidase. So bacterial cells use transpeptidase to basically build cell walls. And what penicillin does is it acts as a suicide inhibitor. That is, it binds into the active site of that enzyme and it begins the catalyzation process, but somewhere down that line, it forms an intermediate that essentially blocks and inhibits the activity of that enzyme, the transpeptidase. Now, aspirin, which has the following structure, is a suicide inhibitor to an enzyme we call cyclooxygenase. And what cyclooxygenase normally does is it basically catalyzes the formation of a special type of signal molecule that is used in the inflammation process. And so when we actually ingest aspirin, aspirin acts as a suicide inhibitor and it binds into the active site of that cyclooxygenase. And so what that does is it disables the ability to produce that signal molecule, and so the inflammation process cannot take place properly. And so that relieves pain, it decreases pain, it removes headaches, and so forth. So another type of suicide inhibitor that we commonly use is AZT. And AZT is a molecule that acts as a suicide inhibitor, and it basically treats HIV. So these are the three different types of irreversible inhibitors. So we have group-specific inhibitors, which are basically these irreversible inhibitors that bind onto specific groups, specific residues, specific amino acids. We have substrate analogs, also known as affinity labels, that essentially resemble that substrate, and so they can fit into the active site of that substrate and modify that particular active site in some covalent way. And we also have suicide inhibitors that basically bind into that substrate, bind into the active site, and they begin the normal process as if they were the normal substrate molecule, but somewhere down that reaction pathway, they essentially create a reactive intermediate that modifies the catalytic residue found inside the active site in some covalent way, and that inhibits the activity of that enzyme. And that inhibits the residue found inside.
This is a substrate analog, right? Homotropic effector.
So for the differences between homotrophic effector molecules and heterotrophic effector molecules in biochemistry, let's start this off by talking about the word origins of each of these. So homo means same, and in this case, these are substances that bind to the substrate site. They can sometimes be the substrate, or they can be other molecules as well. So for an example, oxygen binding in hemoglobin would be a homotrophic effect. The more oxygen you have, the more of those subunits that are going to be in the R state. So if you have more in the R state, the curve itself tends to look more R-like. So we're affecting the shape of the curve by inducing those R state changes. Heterotrophic, as the word hetero means, other or allosteric, for example, would be one of those things. 2,3-BPG or CO2 binding in hemoglobin would be examples of heterotrophic. They don't compete with the substrate at the active site. But what they do is they affect the changes in the Km value. Is it shifted to the right or is it shifted to the left? The Km value, is it shifted to right?
By ATP hydrolysis, these molar proteins are dynein, kinesin, myosin. muscle contraction, vesicle movement, cell motility.
Ciliary dyskinesia. You can see there's some sort of dyskinesia or problem in movement for the cilia, and mutations in a particular dynein protein lead to this rare disease, and therefore an increased susceptibility to chronic enzymatic proteins that we'll be talking about are the antibodies of the immune system. Now, antibodies are protein components of the adaptive immune system, whose main function is to bind foreign antigens and target them for destruction. So in this case, the antigen, which comes from any foreign substance, say a virus or something like that, is the antibody's ligand. So here's an example of an antibody, and then here is the antigen. And the antigen is really just the antibody's particular ligand. So you can think of antibodies as being like little red flags for the body's immune system, letting us know that, hey, this thing is not supposed to be here, we need to get rid of it somehow. And it's important to know that an antibody's affinity for its target antigen is extraordinarily high. So the affinity is strong, really high. And so there you have it. You can see all the different types of non-enzymatic roles or functions that proteins can play, either as receptors or ion channels, as transport proteins, motor proteins, and then highly specific antibodies in our immune system. Okay. Next up are the transport protein in a multi-affinity. And it picks up oxygen in the lungs. So here are my lungs, it's high in oxygen in your lungs, and then delivers this oxygen to tissues. And we'll draw tissue here, say it's muscle tissue, where it's present in low concentrations. And so this is a great example of a transport protein at work. So next up are the motor proteins, which include myosin, kinesin, and dynein, all of which are capable of generating great forces. And these proteins are really crucial for cellular motility. And myosin specifically is a protein responsible for generating the forces exerted by contracting muscles. So every time you flex your bicep, like so, your myosin protein in your muscles are contracting and generating that force. Now, kinesin and dynein are motor proteins that are responsible for intracellular transport. And then dynein in particular also plays a role in the motility of cilia, which are these little extensions of a cell that project out. And mutations in a particular dynein protein can lead to a rare disease called primary ciliary dyskinesia. So in primary ciliary dyskinesia, you can see there's some sort of dyskinesia or problem in movement for the cilia. And mutations in a particular dynein protein lead to this rare disease in which the action of the cilia, the cells lining the respiratory tract, fail to function. And this leads to a decrease in mucus clearance from the lungs, and therefore an increased susceptibility to chronic infections like pneumonia and bronchitis. So as you see, these motor proteins are really important. And then finally, our last class of non-enzymatic proteins that we'll be talking.
Primary ciliary dyskinesia, which transferring of electrons. And one example of oxide reductase would be decarboxylase or dehydrogenase. Alcohol dehydrogenase, for example. Yes. So this NAD plus, because it gets the hydride ion, it is again the electron is reduced, and alcohol is oxidized because it loses the hydrogen. Transferase does what? Amino transfers a functional group, moves a functional group from one molecule to another. Aminotransferase, transaminase, or phosphotransferase. The lyase catalyzes what without the addition of water or the transfer of electrons? It catalyzes cleavage. And an example of that, lipase is one subtype of lyase. Another example would be phosphatase. Oh, decarboxylase, quite really decarboxylase.
Carbon sugar, and kinase is a term referring to enzymes that add. So today we're going to talk about enzymes and all the different kinds of reactions that enzymes can catalyze. But before we do that, let's review the idea that enzymes make biochemical reactions go faster. And if you look at a reaction coordinate diagram, you've noticed that enzymes speed up reactions by lowering their activation energy. Now, enzymes are generally named for their reactions, which is convenient because it makes it a lot easier to remember what an enzyme does if someone gives you its name. And a great example of this is that one of the enzymes involved in DNA replication is called DNA polymerase, which is named as such because it acts on DNA and specifically makes polymers of DNA. Now, the suffix ase is usually just one that you find at the end of most enzyme names. Now, another great example is that the enzyme that catalyzes the first step of glycolysis, which you may remember is the reaction between glucose and ATP to form glucose 6-phosphate and ADP, is called hexokinase. And hexo refers to the number 6, which is a reference to glucose being a 6th carbon sugar, and kinase is a term referring to enzymes that add phosphate functional groups to different substrates. So overall, hexokinase adds phosphates to 6 carbon sugars like glucose. Now, generally, every enzyme has a very specific name that gives insight into the specific reaction that enzyme can catalyze. So we can actually divide most enzymes into 6 different categories based off the kinds of reactions that they catalyze. Now, our first group is the transferase group, and the basic reaction that transferases catalyze are ones where you move some functional group X from molecule B to molecule A. And a great example of one of these reactions occurs during protein translation, where amino acids bound to tRNA molecules are transferred over to the growing polypeptide chain. So in this
This is lysine, Lysk, and it's basic. It's basic and positively charged. This is the Michaelis-Menten equation. Michaelis-Menten. So we have a Michaelis-Menten equation and a Michaelis-Menten reaction. And we also have its graph. So V equals Vmax times S over Vmax plus Km plus S. So the Km is here, the Vmax is here. This is half of Vmax. Substrate concentration is here. And also identificate onto this again. Sigmoidal curve. Cooperative enzymes. Yes. The hydrophobic liquid, the interior of a protein, which increases the entropy of the surrounding water molecules. The less entropy is because an ordered structure. Okay, take a break and I'll be back.